were living S-strain bacteria. He injected dead S-strain, and when he took the mouse apart, he found living S-strain. What Griffith concluded from this work, and correctly, was that somehow the living R-strain bacteria had taken up something from the dead S-strain bacteria. They had incorporated part of the dead S-strain bacteria into them, and that somehow transformed the R-strain into the S-strain. Griffith later showed that you didn't even need the mouse. You could do this in a beaker. If you put dead S-strain and live R-strain bacteria in a beaker, the live R-strain, some of them, would become transformed into live S-strain bacteria. So these bacteria then were incorporating material, and that's an interesting process on its own. But from our point of view, what's really interesting is that the material that they incorporated must somehow be genetic material. It must somehow have information in it. Because how else could the R-strain bacteria now become virulent like an S-strain bacteria? The difference between S and R had to have something to do with genetically transmitted information. So the transformation of the R into S by accumulating or acquiring something from that S must involve genetic information. That's a very interesting finding. What Griffith did was he left it there. I mean, of course, this was only 1929 when he was doing this work. He said that he had discovered what he called transforming principle. The transforming principle in this sense being the stuff, whatever it is, that turned living R-strain into living S-strain that they took from dead S-strain. Our interest in this, and in fact the interest of the scientists who followed this, was that somehow this transforming principle, this transforming stuff, must be genetic material. So all we had to do then is find out what the transforming principle was made of, and we'd have the genetic material. So what kind of molecule was it? Scientists began to ask. Well, in the early part of the 20th century, when Griffith published this work, there was generally an assumption that the genetic material must be a protein. And why did they think that? Well, I mean, they thought it for the reason that we went through last lecture. Pretty much everything that happens in a cell, and they knew this even back then. They had already begun to understand the complexities and diversity of protein function and structure. Pretty much everything that's important that goes on in a cell is done by a protein. So it makes sense that if you've got something complex and important that's being done in a cell, like providing information in this case, then it's probably going to be a protein. Now, they had good reason to suspect even back then that proteins were probably involved specifically from the chromosomal theory of inheritance. Remember, I told you that people knew back then that the way that chromosomes moved corresponded to trait transmission in humans and other organisms. So chromosomes must somehow have the genetic material. So they asked, what are chromosomes made of? And they did some biochemistry, and they found out that chromosomes actually are made of proteins. And some nucleic acids, DNA to be specific. Now, by weight, if you analyze the content of a typical chromosome in a eukaryotic cell, there's 5 to 10 times more protein than there is DNA. So they said, okay, there's a lot of protein in those chromosomes. That's consistent with the idea that it must be proteins. Now, another thing that was going against nucleic acids, DNA specifically, in this context, and we haven't talked about this specifically yet, is that if you look at what nucleic acids, DNA and RNA, are made of, they, like proteins, are polymers that are made up as a string of monomeric little monomer subunits. These are the nucleotides that are all strung together. Proteins, as we've learned, are made up of 20 different kinds of amino acids. Nucleic acids are made up of only 4 different kinds of nucleotides. Another thing that they knew about nucleic acids was that they're actually structurally quite boring. We've already talked in detail about how proteins have these complex structures that determine their function. Well, by and large, and especially back then, nucleic acids just seemed to be strings that lay there. They didn't have these kinds of very complex structures. So the sequence of the building blocks of nucleic acids seemed rather simple, only 4 things. the structure of the nucleic acid, if that had anything to do with its function as an information-bearing molecule, seemed kind of simple and maybe not very useful because it wasn't nearly as complicated as proteins. So everybody assumed that it must be proteins that were somehow holding the code. Now, there was one nagging piece of evidence that argued against the idea of proteins. And this has to do with the fact that if you heat proteins up in general, they break down. They break down because those chemical interactions that we talked about in the last lecture that hold proteins in particular complex shapes start to themselves break apart as the heat rises. And so what happens is the protein denatures, as we say. It actually loses its configuration and stretches out or changes its shape. So literally what that meant was the problem here is that when Griffith had heated up those S-strain bacteria to kill them, he probably denatured a lot and maybe all of the proteins in them, or at least some. So there was some evidence that it might not be proteins, but nonetheless, most biochemists thought that they should be looking at proteins in the early part of the 20th century. So how do you actually answer this question then? How do you go and ask yourself, what is the transforming principle made of? Well, back then, the way to do this was through biochemical procedures that would selectively break down particular kinds of biological molecules. In fact, this kind of work was done in the early 1940s, really quite a long time after Griffith's first discovery, by three researchers at the Rockefeller Institute, what's now the Rockefeller University in New York: Oswald Avery, Macklin McCarty, and Colin MacLeod. These guys were biochemists who had been developing relatively sophisticated techniques at that time for selectively breaking down different classes of biological molecules. Remember, we've got these four major classes to work with that we've talked about. We've got proteins, we've got nucleic acids, we've got carbohydrates, which are sugars and so forth, and we've got lipids, which are fats and so forth. So if what you could do is take a beaker of transforming principle from experiments that were done in the same way that Griffith might have done them, and selectively break down each of these classes of biological molecule, then you could ask which biomolecule, when it's broken down, causes transforming principle to no longer work. So, I mean, this is the way you do the assay. You have some sort of transforming assay with things like S-strain or R-strain bacteria, although by that time many other kinds of bacterial systems were being used. You extract from a solution some substance which you would call transforming principle if you were Griffith, and then you treat that solution biochemically in selective ways to selectively knock out the proteins or the nucleic acids or the carbohydrates or the lipids. And then you ask which one, when it's broken down, ends up causing transforming principle to no longer transform. So they did that, and here's what they found. They could break down the carbohydrates, no problem, still got transformation. They could break down the lipids, no problem, still got transformation. They could break down the proteins, and there was no problem, they still got transformation. But if they broke down the nucleic acids, the transforming stopped. And they concluded from that, and I think in a very straightforward way, that the transforming principle that Griffith had discovered therefore must be some kind of nucleic acid. Now, to me, that's pretty good evidence, but interestingly, in the 1940s, that result wasn't widely accepted, not for a couple of reasons. First of all, there was growing interest in protein biochemistry, and a lot of people were still focusing on the importance of proteins. So there was a bias against believing it could possibly not be proteins. The other reason that people were critical, which is a fair criticism, is that the biochemical techniques that these researchers were using were relatively novel techniques, and there was some argument that maybe they may not have destroyed all of the class of molecules that they thought they destroyed. Specifically, there was some suggestion that when they thought they broke down all of the proteins in one of these solutions, they didn't get them all.